All right, hi everybody. Welcome to June Shrestha's thesis defense. I'm Scott Hamilton, her major thesis advisor. So before we get started, I want to thank the Moss Landing IT group for getting the virtual thesis defense all set up and going. And I want to remind everyone to please keep yourself muted. I think we'll just force that, but you'll be muted throughout the presentation. And then at the end of the presentation, you'll be able to ask live questions to June. And so you can use the raise hand feature, which is under the participants tab at the bottom of your screen. You'll be able to find that feature. Raise your hand and then we'll call on you, we'll unmute you and allow you to ask June some questions. And then after that's over, we'll let everyone congratulate June, and then we'll ask you all to leave, and then her thesis committee will stay along to ask a few more questions. So yeah, we'll just do a quick little introduction here to tell you a little bit about June and her background. As many of you know, June is a very special person. I know she's touched many of you at her time here at Moss Landing. It's been 5 years to finish this thesis, thereabouts. But really what you'll see, June has not been slacking during that time. She has been very active, working very hard on what I think is a very interesting and really groundbreaking thesis. In addition, as you'll hear, June has really never met an opportunity that she did not like. She was always helping out all of her lab mates with their experiments and projects, just taking advantage of different internships and other exciting things that came along. I probably did a poor job discouraging her from doing those things, but I think she's really grown a lot in the process. So let's talk a little bit about June. So I think, doing a, here we go, right? Doing a master's in marine science was definitely a good fit for June. Right, she's always lived a life aquatic, been in and out of the water. So here's an example with her and her sister, early alligator tamers. Right, she's always been a competitive swimmer ever since she was young, all throughout high school, maybe through college as well, June, I can't remember.
Yeah, on a club team.
Yeah. Right, at Moss Landing, she became a seasoned boat operator to do all of her research, driving small vessels all over the place. She's also a fish collector extraordinaire. As you hear in her research, she had to use pretty much every single technique I think that's ever been invented to try to catch live fish, from nets to hook and line, above the water, underwater traps. Here she has, 10 different collection items attached to her as she's going about to do her job. Right, June loves to travel. She loves to spend time with her friends and family. She especially loves to spend time out exploring nature and having all sorts of great adventures. We affectionately like to refer to June in the lab as Mama June. So June has a very nurturing, right, and caring personality. Right here, you can see to all different life forms, right, especially her fishes. But she's kind of one of the ones in the lab that would always kind of step up, right, and do the things that no one else really wanted to do, and just get it done. She would organize the lab cleanups, maintain the chemical inventory, take care of the lab calendar. She was helping with all the party planning. She was always volunteering to help her lab mates, always doing things selflessly. And we really thank her for a lot of that. So let's see, is there a bit more evidence out there for this Mama June character? Well, like any good parent, right, she's always concerned about people's health and well-being. So here, very important, trying to keep people hydrated. I know I'm doing all the same with my kids all the time, yelling at them to always have a drink.
I thought what happens in Mexico stays in Mexico.
Except for introductions. Right? She also wants to make sure before you exercise, right, you got to do your stretches there so you don't pull a muscle. Right? She's always concerned making sure that you're getting well rested, even aggressively so, telling you make sure you get your 8 hours of sleep. Right? Mama June, she's also a committed educator and a mentor. She was always seeking to steer others in the right direction. So she serves as a teaching assistant for 3 of the courses here at Moss Landing. So our Baja class, the first time we did a trip down to Isla Natividad, where she helped students do all of their independent research projects and help plan all the logistics for a trip to a new location, very remote location. She was the TA for the marine ichthyology class and the marine ecology class, again, helping students with independent projects, revising lab manuals, and really making the class run much more smoothly, and especially setting up future TAs so their job would be better. She was also a very committed mentor. So 8 different students worked with June, helping with her on her research projects, but she also spent a lot of time mentoring them on their own independent research projects, setting them off in the right direction, and spending a lot of time helping with them with presentations and posters and all sorts of things. All right, some other things you may not know about June. She does like to strike a pose, right? Make sure you know she's been there, right? She trained it. If this career in marine science doesn't work out, modeling is always an option. She also sets new fashion trends wherever she goes. She's one of those fashion or social influencers. So here's just a few of her fashion trends that I really like. One, double glasses, as you can see here. Here's my favorite, underwater rain jacket, right? You don't want to get wet. You got to protect yourself. And one of my other favorites, indoor sun hat. So watch out. These fashion trends are racing across the nation. Right, some additional fashion trends, and if you've been with June in the field, you'll see she has a pretty strong aversion to sunlight. So I'm still not sure, are these fashion trends, or maybe she's secretly a vampire? I don't know. Right, as you'll hear in her research, again, as I mentioned earlier, she's an expert fish collector and urine sampler. So that's kind of been her life for the past few years, is sampling fish pee. Right. And she's been very successful in all her research endeavors. I can imagine here in this photo, this is probably a vat of concentrated fish pee that she's about to analyze. Right, we have some evidence at the bottom here from her first science fair project. I'm not really sure what that contraption is, but again, I'm sure it was a revolutionary discovery that you made. She's also been very successful in getting grants to support her research. So here you see a long list. She's raised over $20,000 to support her research. And been, again, very successful that way. Right, she's also always looking to the future. She really strives to make a difference, and so she's gotten involved in a variety of policy activities. So she spent a summer doing an internship with the Marine Sanctuary Office here in Monterey, where she wrote a 100-page report looking at the linkages between national forests and international marine sanctuaries. She also developed and hosted a workshop with the marine groups, which is for grad students throughout the Monterey Bay Area, on linking science to policy. She's also very committed to outreach and service. So here again, you can see a long list of many of the other activities she's been involved in. So a few highlights: she's been a tour guide for a long time, right, showing groups all around Moss Landing and telling them about the lab. For 2 years, she was on the student committee at the Western Society of Naturalists, helping to organize a lot of the student activities and events at that conference. She was the co-chair of the Moss Landing Open House for 2 years, which is just a monumental undertaking. And that's where we open up the lab to the public and welcome a couple thousand people over a long weekend. And then in her free time, if she didn't have any extra free time, right, she also volunteers at the Monterey Bay Aquarium as a diver in the kelp forest tank, where she gets to don one of those cool face masks and interact and answer questions from the public. So last but not least, June's other skill, you might know that she is a gifted musician, right? She is a star, as you can see from this photo from when she was a young child. But then also I have some video evidence from a recent Christmas party at my house.
Oh.
Good times. So yeah, but last but not least, so right, we're saying goodbye to June, right, and she's saying goodbye to Moss Landing, right, and hello to some new adventures. And starting now in just a couple of weeks, she's going to be starting a California Sea Grant State Fellowship with the Channel Islands National Marine Sanctuary. And so we're really excited for June to move on to this next phase of her career. All right, so with that, I'm very excited to introduce June, and she's going to be telling you all about the fascinating world of fish pee. All right, you should be able to share now, June.
Okay, we in business here?
You're in business. Good to go.
Awesome. Thank you for that introduction, Scott. That was amazing, and definitely some footage in there that I'd totally forgotten about. I'm really excited to be here speaking with you all today about my research from the last 5 years. where I studied fish pee in the sea as a hidden source of a limiting nutrient for California kelp forests. I'm happy to see so many of you able to tune in on Zoom today. And I think one silver lining with the COVID reality is that so many of you are able to tune in from across the country, including some of my family on the East Coast, as well as previous researchers I used to work with as an undergrad. So I think it's really exciting to be able to come full circle here and tell you about my work. So I have you for about 45 minutes where I will be sharing my research with you. Let's see. There we go. So a quick little overview of how the presentation is going to go. I have, oops, sorry, give me 1 second. There we go. I have to close all these windows here. Okay, so first I have my introduction, and then I'll follow that with a discussion into my research questions. I have 3 different primary questions, which I then split into 3 parts for the bulk of my talk. And in each of those parts, I'll discuss the methods, results, and the discussion for how I answered that question. Then I'll wrap it up with some overarching conclusions and my very important acknowledgements. So today I'm going to be talking to you about ecosystem ecology. Thinking big picture, all of the ecosystems on Earth generally require 2 things to function, and that's the provision of energy and nutrients. Energy can be derived from the sun and is transferred through the food web via predator-prey interactions. Nutrients, on the other hand, are cycled throughout ecosystems and are needed to support development in living things. And so my research over the last 5 years asks a fundamental question in ecology, and that's, what are the roles that animals play in an ecosystem? And how can animals help support these key ecosystem processes? So many of us might be familiar with the role that animals play in helping to provision energy to an ecosystem. It's the idea of how primary producers convert energy from the sun, and that energy is passed up through the food chain as predators eat their prey. However, animals may also help to mediate nutrient cycles within an ecosystem. They can do this in 2 ways. The first being when they die and their body is broken down by decomposers, and the nutrients are returned back to the earth and then once again utilized by the primary producers. However, animals can also help mediate nutrient cycles even when they're alive, and that's through their pee and their poop. Yes, it's a scientific terms, peeing and pooping. And so, although the energy-driven approach to looking at trophic cascades is what dominated early literature, ecologists now recognize that animals in many ecosystems are important nutrient contributors. For example, it's been studied with plankton in the open ocean, mammals in terrestrial systems, and fishes in lakes, streams, and on coral reefs. So to hopefully answer some of your burning questions about how animal excrement works, basically, if you have a consumer, they eat food, store the nutrients they need, and excrete the rest. And hopefully this process isn't too foreign to you because people, we eat and excrete in a very similar fashion. So in fishes, I wanted to focus on what we know so far about fish storage and excretion of nutrients because they're my study species. And they can store usually about 30% of the nutrients and release up to 70% of what they're eating. But the rates at which they store and release the nutrients, it varies depending on things like their taxonomy, as well as their body size. And fish, they are actually constantly excreting nutrients. They release it through their gills and their urine. That's what I actually kind of call the fish pee. It's any dissolved nutrient. And then the poop is just a minority of the way that they release nutrients, so less than 20%. So for the purposes of my talk, I'm not considering their fecal matter and just focusing on these two, the dissolved nutrients. But together, no matter if it's coming from their gills or their kind of urine, they're excreting ammonium, which is NH4+. Ammonium is a source of nitrogen. So now that takes me to kelp forests, my study ecosystem. So this photo, it's of a kelp bed in Southern California, taken from a scuba diver's point of view. And in it, we can see those long green stalks. That's giant kelp, Macrocystis porifera. Giant kelp is super impressive. It's the fastest growing photosynthesizer in the world. They can grow to be 50 feet plus tall. And these ecosystems occur all over the world. So the type of kelp forests that create that tall structure, like I just showed in the picture, can be found in the red areas on this map. And in California, we have beautiful kelp beds nearly all along our coast, as indicated on this map in gray. So giant kelp forests really sustain one of the most diverse and productive ecosystems on the planet. And honestly, when I'm scuba diving down there, it's so beautiful. There's so much to look at. And in it, the food web is complex and primary production is extremely abundant. Our own Dr. Mike Graham, Dr. Evil, some might say, has reported on many of these predator-prey interactions that can be found in kelp beds. And sometimes even thesis advisors can be partaking in some of its delicacies. So it's important to have a well-rounded understanding of how nutrients cycle within kelp forests, and especially how nutrients are utilized by giant kelp. As a foundational species, giant kelp provisions food and shelter for the whole ecosystem, so its loss could really be detrimental to the system. So it's often assumed that upwelling is the sole mechanism to deliver nitrogen to kelp forests year-round. Upwelling, it's an oceanographic process that supplies nutrients, mostly nitrogen, to the coastal zone. It's when the wind-driven movement of cooler, nutrient-rich, deep water gets pulled towards the ocean surface, which is replacing the nutrient-depleted water that gets pushed offshore. However, the problem with this paradigm is that upwelling is not consistent year-round, and it's seasonally variable. And with the predicted effects of climate change, it's likely that the delivery of nutrients from upwelling will decline. So then the question remains, how does giant kelp maintain biomass year-round during low nutrient periods? So here I have a picture of what some kelp might look like in the spring and into the early summer months. An individual giant kelp individual will have a lot of biomass during this time. And this is also when upwelling is very strong in the spring. But then in the fall and into the winter, nitrate availability drops and is consistently below giant kelp's growth threshold. Upwelling can be reduced up to 80% in this season. Yet kelp biomass persists, and it might be reduced. It might not look as full as in the high productivity months, yet it persists. And giant kelp, it cannot store nitrogen very well. Thus, it's been proposed that giant kelp needs multiple sources of nitrogen to sustain year-round growth. So we know that oceanographic processes might supply about 70% of the nitrogen needed. And that lasts a little bit, maybe the animals are supporting their last needs. And animal pee, it's rich in ammonium. Again, it's the NH4+, whereas the nitrogen type from the oceanographic sources are usually nitrates. And what's interesting is that the nitrogen type supplied by animals is a different type. Ammonium is actually typically the preferred type of nitrogen utilized by primary producers, and because it's more easily utilized. So with this immense potential for animals to help supplement the nitrogen pool, it's kind of surprising that animal pee in kelp forests is not yet well studied. What we know stems from 3 papers. The first one tested the excretion rate by blacksmith, which is a schooling and very dense fish species typical of kelp beds in Southern California. In that study, they also found that giant kelp does utilize the nutrients from blacksmith, especially when they're sheltered at night in the crevices of the individual kelp. And we know a little bit more about the role of animal nutrients from invertebrates. We now know that excretion rates for the majority of the invertebrate community, we know the differences for these different taxa, and also how the magnitude of their excretion responds to warming events and anthropogenic impacts. But in addition to those consumers, there's over 30 more fish species that reside within the kelp beds. So there's really a lot of more work to be done. And that takes me into my research questions. My first research question was just to quantify how much ammonium do kelp forest fishes excrete? What's their individual pee rate? My second question asked, what is the excretion rate of ammonium from the whole community? And then the third, how does fish pee compare to the other sources of nitrogen? So I've split these 3 questions into 3 parts, which I'll focus on for the rest of my talk. And for each section, I'll go through the methods, results, and a brief discussion separately before moving on to the next part. So here we go, part 1. How much ammonium does an individual fish excrete? So my goal here was to ultimately generate some predictive ammonium excretion equations that I could use to answer the questions in part 2 and 3. So I wondered, do all fishes excrete ammonium at a similar rate, or does it vary by taxonomy and fish size? These variables were deemed important with fishes in other aquatic systems. So now I'll share a little bit about my methods and how one goes about measuring fish pee. Oh, and this is a picture of Scott, my advisor, and one of my amazing volunteers, Colette, in our little field research station. So a quick overview of the process, and then I'll go a little more in depth with each step. I had 2 field seasons. They lasted from June through September in 2016 and 2017. I sampled fishes in both Central and Southern California in order to capture a wide variety of fishes that associate with kelp forests. And those sites in Central California, as I went to Stillwater Cove and Point Lobos, the more northern star on the map, whereas the Southern California site was sampled in the Santa Catalina Island in the Channel Islands. So then to actually measure the fish excretion rate, I conducted short 1-hour experiments to measure the amount of ammonium they excreted over time. And that's how I calculated the rate. Okay, so first, the fish collections, maybe arguably the most fun part. So we went out and collected over 500 fishes from 30 different species. And I collected at least 10 to 30 individuals per species. And I did that because previous studies indicated that 10 to 30 individuals was sufficient to generate a strong relationship between size and excretion. I also only kept and used fishes that were in good condition, so not showing signs of barotrauma. We used a variety of gear types to capture species across their size range. So some of the types we used these fish traps, hook and line surveys, as well as scuba diving techniques like what Scott alluded to. Those are really fun. That involved setting up these giant barrier nets underwater and then kind of corralling the fish towards the net and capturing them. And preliminary tests I did indicated that the gear type I used did not influence the excretion rates. So then once I had the fish in hand, we set up the experiment because we did the experiments in the field. So for the incubations, I placed individual fish into separate tanks. I was very methodical with this process. Each tank was treated the same. It had a bag. Each tank was lined with a plastic bag that was filled with seawater. The seawater was filtered to remove the bacteria and plankton that might otherwise influence nutrient concentrations in the sample. And it was amazing. By the end of the process, my team and I, we could do up to 10 incubations almost simultaneously, kind of just staggered slightly, as depicted in this picture on the right. And oh, I should mention that I did correct for the water volume in each of those tanks later during the calculations. So then to conduct the experiment, I incubated each fish for 30 to 60 minutes and collected a water sample every 10 minutes. Now, I ran the experiments for 60 minutes as I was unsure if the excretion rate would vary throughout the experiment. But with fishes and other systems, it was deemed that 30 minutes was appropriate. But I just wanted to do my due diligence. And so after I ran incubations on at least 10 individuals of each species, and I saw that the rate did not change over the duration of the experiment, I then reduced the time to 30 minutes. So then after the experiment, that's when I weighed and measured the fish for their total length. And what I loved about it is I was then able to release the fish back into the water. So my only samples were those vials with the seawater in it. So at the end of our field day, after we all scarfed down some dinner, that's when we analyzed the seawater samples for the nutrient content. For the ammonium analysis, it works very similarly to like a home aquarium ammonium testing kit, where basically when I mixed the seawater sample with a working reagent, after some time it would fluoresce and actually change color. And then when I put the sample into a fluorometer, the fluorometer could pick up the exact concentration of nutrient in that sample and put a number to it. And that is resulting, or enabled me to calculate my results to see how excretion increased over the experimental time in the bucket. Okay, so that leads me to the results of this section. This is a great picture of 3 of my interns that worked with me in 2017. Here we're sampling in Central California. And Marissa in the back, she's holding one of the little pipettes that we used to collect the seawater samples. Okay, so ammonium, this graph shows the ammonium concentration in each sample. And so on the y-axis is the excretion from the individual fish, and the x is the elapsed time in minutes. So each point here basically represents a different sample. And so the excretion rate then was basically just the slope of this best fit line. And there was a cumulative excretion went up throughout the duration of it, which is good. So it means that they weren't holding their pee in or something. And we saw a similar trend for all 500 fish. So I took the slope from this linear regression and plotted it as individual points on this following graph. So here this graph has, again, the excretion on the y-axis and fish mass, the fish's weight, on the x-axis. Both of these were log transformed due to evidence of allometric scaling. And so again, each of these points were the different slopes. So there's the 500 fish represented here. And each color of those points represents a different family the fish was from. So examining the best fit line here for this linear regression indicates that excretion rates scaled allometrically, which is just another way of saying that the bigger fish actually excreted ammonium slightly slower than the smaller fish. So if it was equal, then that slope, which is this number up here on the equation, that would have been pretty much at 1. And this result is frequently seen among fishes in aquatic systems. And so just another way to kind of visualize that, what I mean by that is if you're controlling for body size and you have 2 fish that are actually the same exact weight, then it's actually the smaller fish that would excrete more ammonium than the larger fish per gram of weight. So in the wild, you might see a situation like this if you're in an area maybe with a lot of juvenile fishes, or if you're in a popular fishing spot where all the bigger fishes have been harvested by fishermen. Conversely, if you allow the fish size to propagate through, and this is kind of more typical of the actual ratios of what the fish look like in the wild, then the bigger fish per individual would actually excrete more ammonium than the smaller individual fish. So returning to this graph, I also tested if excretion rates varied by family and keeping the fish mass as the covariate. So here, now each line represents a different family excretion rate. And since the slopes of these lines differed, I concluded that the excretion varied by family. Thus, for the next 2 sections of my research, I used excretion equations that were specific for what family the fish was from. Okay. So now to wrap up this part 1, to compare the mass-specific excretion rates to other fishes, it's actually quite comparable. It's in the same range. And interestingly, the excretion rates are also very similar to invertebrates in kelp forests. Okay, wrapping up part 1, how much ammonium does an individual fish excrete? Well, on average, if you had to put a number on it, it's about 1 micromole per gram of weight per hour. I learned in this section that the excretion, it varies by body size and family. And thus, I was able to generate those predictive excretion equations. And what I think is really powerful about generating those equations is that we can pair them with any study of kelp forest regions of interest, which I think is really powerful in order to be able to look at other regions as long as the families are somewhat consistent. Okay, moving into part 2 of my research, how much ammonium does an entire community supply to a kelp bed? So here, now I've wrapped up my empirical study and now I'm moving into some modeling where I estimated the excretion by all fishes in a community, taking their densities and sizes into account. And it was a great way to see some of the community level excretion trends because I wouldn't possibly be able to conduct empirical experiments on literally every fish in a kelp bed. So I chose to study the Northern Channel Islands as my case study, which is located in Southern California, just northwest of Los Angeles. The Northern Channel Islands They are comprised of 4 islands. From west to east, you have San Miguel, Santa Rosa, Santa Cruz, and Anacapa. Although it's a relatively small geographic area, you can actually travel by boat between these islands in one day. There's a really interesting environmental gradient that supports a diverse community of fishes here. The islands sit at the confluence of 2 major currents, with the cool, nutrient-rich current bathing the western islands from the north, and there's a warmer countercurrent encompassing the eastern islands from the south. And this results in substantial variation in productivity as well as fish community structure. In addition, the Northern Channel Islands, they hold a lot of value for people. They're popular destinations for recreational fishing trips. And here are some of the species that are targeted recreationally, including lingcod on the Western Islands, as well as California sheephead and kelp bass on the Eastern Islands. So my questions for part 2, I asked, does community excretion rates vary spatially and or temporally? And what factors drive the variability? So to give a brief overview on how the modeling worked, basically I calculated the ammonium which would have been excreted per individual in that area. I then used the mass-specific predictive excretion equations. Again, I generated that in the last section. Those individual rates were converted on an areal scale by multiplying by the density and size of all fishes observed in that area. And then I analyzed it all for the spatio and temporal trends. I did this at 46 independent sites across 14 years of data. The estimates of community structure were conducted by PISCO, which is a great long-term kelp forest monitoring program. And basically the divers at each site, they conducted multiple fish transects at different levels within the water column and conducted in a stratified random design. Those on the fish surveys, the divers identified, counted, and estimated the fish length of all fishes observed in a standardized area. And I just think it's incredible that over the 14 years of data, their divers counted over 140,000 fishes. And so I was able to predict the excretion rates for the majority of them, and it represented 95% of the total fish biomass. And on those surveys, it was really important that the divers estimated the fish lengths, because as we saw in part 1, the fish P rates scale with size. So I converted the fish lengths to weights using established equations. And my advisor, Scott, he was a researcher with PISCO at the time, and I learned that he was just so committed to collecting good data that he'd even practice sizing fish while above water, as evidenced by these photos. And so I don't even want to think about how much ammonium or nutrients Scott was contributing to those dive surveys, but that's a study for another time. So now I'll share some of the results from part 2. First, I looked at the results for the question, does excretion vary spatially and or temporally? I observed that areal excretion rates did vary spatially. It varied not only by site, but also by island and reflective of the overarching fish community structure. So I created this map in ArcGIS. We have the 4 islands, and each of the bubbles represent the different sites. And so the bigger the bubble and the darker the color meant that there was a greater delivery of nutrients. And the units here, it's micromoles per hour per meter squared. So the average across all of these sites came out to be about 131 micromoles per hour per meter squared. So I also wondered then, did aerial excretion rates increase over time? And yes, it did. So here I have a graph depicting the mean excretion rates on the y-axis, and then the year of the different sample years on the x-axis from 2005 through 2018. So the excretion rates, they generally actually increased over time. Leading me to my next question, what factors drove the spatiotemporal variability? And I had two ideas here. First, I thought it might be due to the effect of marine reserves, as well as maybe the effect of environmental conditions. So first, I thought that marine reserves might be at play because it's well documented that fishing can reduce fish population sizes. In the Northern Channel Islands, fish abundance, diversity, biomass, and size is frequently greater inside of the MPAs than outside of them. And this trend is especially strong among the species targeted by recreational fishermen. So I wondered, do nutrients follow a similar pattern? And I hypothesized that I would see a similar trend with fish excretion. So I overlaid on the same map you saw before, but now I added a layer showing where the different reserves are with each site. And yes, my hypothesis was supported, and aerial excretion is greater within marine reserves. So most of these sites with the larger bubbles do lie within the protected areas. And actually, if we look at the average, the aerial excretion rate was about 25% greater inside of marine reserves compared to outside of them. The temporal excretion rates are also correlated with the establishment of marine protected areas. So in the Northern Channel Islands, the reserves were established in 2003. And there's the two lines here now, the averages based on if the site was inside the reserve in red or a site open to fishing, so outside the reserve in blue. So we can see this increase over time, especially with the sites inside the marine reserves, which is likely due to the recovery of the targeted species after the restrictions on fishing were put in place in 2003. So in addition to the effect of marine reserves, I wondered if excretion patterns were correlated with the environmental characteristics. I asked this question because sites around the Channel Islands can be quite variable. As a reminder, there's this mixing of currents resulting in various exposures to sea surface temperature, wave action, and habitat. So to answer this question, I used stepwise model selection techniques of 13 different environmental variables to test if spatial excretion was correlated with these environmental factors. And the best fit model indicated that 70% of the spatial variation could be explained by these habitat and environmental characteristics. It was positively correlated with these variables on the left. And it really just indicated that in locations with lots of giant kelp biomass, lots of topography, and high primary productivity in the water column, that's what supported more excretion. It was negatively correlated with these three variables, thus indicating that excretion was greater in areas where the understory kelp density was sparse, the site was protected from waves, and interestingly, the relatively new metric, the distance to the shelf break, indicated to me that as the sites closer to this drop-off point essentially had greater excretion, and I think this is indicative of the greater delivery of oceanographic nutrients to those sites. So similarly, I wondered if some of the temporal variability that I observed could be correlated with the environment. This is the same graph you saw before, the excretion rates over time. Because in the Northern Channel Islands, the ocean was changing during the time series. There was a documented set of heat waves, the first one in 2008 and a subsequent one in 2014 to 2016. There was also a period of lower than average salinity, nutrients, and chlorophyll, as well as a recorded El Nino Southern Oscillation climatic event. So I thought that maybe some of these things could help explain some of the temporal variability in excretion. So following similar stepwise model selection techniques I used to examine the spatial patterns, I found that the environmental change explained 57% of the temporal variation. And the first variable included in the best fit model was a climate indicator, this NPGO, which stands for the North Pacific Gyre Oscillation Index. It's a bit of a mouthful, but basically the NPGO index is indicative of circulation patterns in the North Pacific, and it describes correlations between salinity, nutrients, and chlorophyll with sea level height. The second variable in the best fit model actually was the density of fish recruits to the kelp beds. So fish recruitment, you can think about it as fish babies kind of settling into the kelp forest from outside areas. So for both of these variables, the best fit was when the variable is offset by 2 years, which kind of makes sense because it would take 2 years to see noticeable effects on the fish community, especially something like fish recruitment, where it takes time for the fish to grow bigger and then start to be excreting more noticeable levels of ammonium. All right, so we're at the end of part 2, and I just wanted to summarize my results here. My first question I asked, does aerial excretion rates, do they vary spatially? Yes, they do. Do aerial excretion rates vary temporally? Yes. Is aerial excretion correlated with the establishment of marine reserves? Yes. And is aerial excretion correlated with environmental factors? Yes. Very exciting here. So now I'm going to move into part 3, the third and final section, and that is where I asked, how does fish pee compare to other sources of nitrogen? Not just other sources of nitrogen, but other aquatic consumers as well. So this section is not as long and there are not as many graphs. So first, I compared aerial excretion rates just to fishes and other ecosystems and found that excretion is generally about the same as excretion in tropical reefs, like the middle picture, as well as in freshwater systems like the one on the right. The ranges you see for those other numbers is just indicative of what the overarching community structure in those systems look like. And then comparing fish community excretion to invertebrates, the aerial excretion is actually 6 times greater among fishes than among the invertebrate communities. So this is a little bit of an estimation because the study that looked at the invertebrate excretion rates, they were done on the mainland in Santa Barbara. So the communities are a little bit different. And at their peak rates for the invertebrates, they were at about 18 micromoles per meter squared per hour, whereas the fishes were at about 130. And I think that this is, although the mean mass-specific excretion rates were relatively the same between these species, it's because the fishes can grow to be so much bigger than invertebrates that on a whole they excrete much more, thus the aerial estimates are larger. So this is my last graph I'm going to show you, and it's the way I looked at comparing fish pee relative to other sources of nitrogen in kelp forests. So this is basically all we've known about comparing these rates. I put the nitrogen supply on the y-axis here. This I calculated is the micromole per liter per day. So it now is on a volumetric scale. So I had to convert the estimates of excretion from the animals for both the fishes and the invertebrates. I had to convert it to this volumetric scale. However, the other oceanographic estimates, the upwelling bars and the internal waves bars, that was already ready to go. And so now comparing, we saw, yes, the fish, they supplement these other sources of nutrients, and they're actually comparable to the amount of nitrate delivered from the internal waves. And it's, you know, it's not exceeding the amount from the upwelling, and I never thought I would necessarily exceed it. But what I think is really important to recognize is that the fish pee in yellow on the two right bars, they're giving this localized source of nitrogen that, because they're constantly peeing through their gills, basically, it's staying within the ecosystem. Whereas the nutrients coming from these oceanographic sources, it's very dependent on those delivery rates and might be variable with changing climatic conditions. I'll also mention that I calculated the estimated ammonium also for those oceanographic delivery sources. So that's what we see in yellow at the bottom on those left three bars. And so the fishes were really the primary source of ammonium. Okay, so we've wrapped up my 3 sections here, and I just wanted to summarize some of my conclusions and my main findings. So from part 1, I measured the excretion rates for over 30 species of fish and found that excretion rates per individual depends on the family identity and size of the fish. In part 2, I found that aerial excretion rates vary by community, and excretion rates are correlated with marine reserve effects and environmental conditions. Lastly, part 3, the main takeaway here is that fishes do supply a supplemental source of nitrogen, and they're the highest source of ammonium. So really, there's so much more I wish we could talk about, but in the interest of time and probably everyone's sanity, this was really just the key takeaways from my research. My work was split into 2 chapters, and so I encourage you, if you have any more questions about all of this, to read my thesis, but also I hope to publish both of these chapters as separate publications in the coming year. So hopefully it'll be out soon. So the first publication would focus on really digging into the empirical results and looking at the different factors that influence the ammonium excretion rates per individual fish. Whereas the second publication would dig into the aerial results and the results from the Northern Channel Islands some more. And I also just wanted to put a little plug in for the fact I also, not presented here, I also measured phosphate excreted by the fish community. So I didn't talk about it here. I also haven't analyzed all of that data either. So if there's anyone listening to this who's maybe looking for a thesis project, I have data we could share. So talk to me. Okay, so then lastly, I just wanted to wrap up with some of kind of my main takeaways about why I think we should really care about fish pee and kelp beds. So the fishes, they're supplying a previously unquantified source of nitrogen. The fish, they fertilize their own habitat, and thus help to increase primary productivity right where they live. The fishes, they help increase kelp forest resiliency. The potential to do this will become especially important in the future as predicted climatic conditions might change the rate where upwelling and other sources of nutrients are delivered. And lastly, the fishes, or my research, really supports the idea of having ecosystem-based management approaches, because recognizing that spatially protected areas may help protect some ecosystem processes, such as nutrient cycles, which are otherwise hidden at first glance. So you all made it. Thank you so much for listening. I just wanted to take a few moments to acknowledge all of the people that really made this work possible. So firstly, a big thank you to my committee members, Dr. Scott Hamilton, Dr. Rick Starr, and Dr. Kenneth Cole. So Scott took me into his lab. He's my primary advisor, and starting in 2015 with him, I feel like I've just grown so much under Scott's guidance and being in his lab. I've grown as a scientist for sure, also as just an overall human being. It was amazing to be in his lab because he's so approachable. You know, I would frequently unannounced just stop by his office and we'd chat about fish pee. But he's also amazing to learn from in the field and frequently came out with me sampling. He made all of the Southern California sampling. He really made that happen. And so I'm really grateful to Scott, really grateful for the opportunity to have been in his lab and accepted all of those years ago. Thank you, Scott. I'd also like to say thank you to Rick Starr. He's the PI from the Fisheries and Conservation Biology Lab at Moss Landing. Rick is a fish genius. And so I learned so much about fish biology and thinking about fishes from him. I also learned a lot about writing with Rick. I took one of his fisheries classes with Scott in my first semester. And Rick, he really took me under his wing when I first got to California, and he kind of adopted me into the fisheries lab a little bit. So I was really grateful to have that expanded community. So thank you, Rick. And then a big thank you also to Dr. Kenneth Cole. Kenneth is so smart and all of the things biogeochemically related. So I kind of came into this thesis from the fish perspective and had a lot of learning to do about placing the nutrients in context with the chemical cycles. And amazingly, chemistry was not my strongest forte as an undergrad. So I had a lot of learning to do. So Kenneth really also took me under his wing and was so great to learn from, and is really so strong at thinking big picture here, thinking big processes for the ecosystem. He was also really fun. All three of these advisors, I went out in the field with them. They're all so fun to be with around in the field with. So thank you, Kenneth, and thank you, everyone. So I have a few more acknowledgement slides. I'd like to say a big thank you to all of my interns, and especially thank you to these three, Cameron, Joseph, and Marissa. They were my UROC and REU interns in 2017. And we spent literally every day together for like a 3.5 month period. And there were some really long field days, and they were just so great to be around in the field, had really positive attitudes. And it was so fun to also kind of feel like we had our own little mini lab, because it was really fun to learn how to mentor them as well and see how they have really grown in their science since 2017 as well. A big thank you to all of my friends who helped with field and lab work. It truly took a village. I circled your names on the left because these are the guys that came out with me for some of my longer sampling periods. So for example, like Amanda, Kenji, and Emmett, Heathers, they all came down to Catalina with me as my dive support team, and they were helping me catch fish underwater for like a 2-week period. Whereas Colette and Nick, Joe, Alex, all these guys were helping more on the nutrient side and helping me run the experiments on the surface, which was also super critical. It was so great to have this consistent help, this support from these guys. But also I had help from so many other people from Moss Landing. Your names are on the screen, and I'm so sorry if I forgot your name on here. The field days were such a blur. They were so long, and thank you for working so hard in the field and in the lab to help me collect these data. And even that picture on the right, that's Hudson, Scott's son, was even helping me catch fish. And I have a big thank you to give to my friends and family. This is just a small subset of everyone. I'm so grateful for you to helping me to stay sane and, you know, keep it light and keep it fun. And we had a lot of really good times over the last 5 years, as evidenced in some of these pictures. And a big thank you to my family there in that left corner there. That was from a couple of Christmases ago. And just really so many more people out there. This is just a start of the list here. Firstly, the academic support, I really want to recognize Joey Peters at UCSB. Joey, we kind of crossed paths a little later on this nutrient journey than probably would have been helpful to start earlier, but Joey is the one working on the Invertebrate P essentially in Santa Barbara. And he helped me with a lot of the modeling that you saw in the second part. So I really could not have done those aerial estimates without him. I also like to thank Tom. He helped me, Tom Connolly, he's the physical oceanographer at Moss Landing. He is the one that helped me understand some of the oceanographic models and how to get data for that. And then a big thank you also to Kim. I don't know if Kim's in the Zoom meeting today, but Kim was in the trace metals, trace nutrients lab, and she helped me learn so much about the nitty gritty of how to collect accurate data and, you know, differences between vial types, which pipettes I should use in the field. She also ran some of my samples because I analyze everything manually, but she has this big fancy machine in the lab there. And so she would, for free, run some of my samples to make sure like the numbers were matching. So I'm just really thankful that she didn't charge me to do that and that she offered, and such a great source of support. And at Moss Landing Marine Labs, really like to thank the people in the ichthyology lab and fisheries conservation biology lab. They provided a lot of support in helping me design and develop these ideas. And they supported me with lots of fun times as well. And in the chemical oceanography lab, a lot of those guys helped me with analyzing some nutrients. And the phycology lab, again, I came at this research from a very much a fish approach. So I had some learning to do about kelp and uptake rates and everything. So these guys helped me place everything in context within the giant kelp ecosystem. Oh. And then the staff at Moss Landing Marine Labs, it's such an incredible community at the Marine Labs, Marine Ops team, like JD Jackson, everyone. They were making sure I was staying safe in the field, that I wasn't going to flip the boat or something. The IT staff, Michael and Brett, and the help crew frequently had computer problems, and they would just fix them. It was amazing. The library staff, including Katie and the other library support team. Front office, Michelle, Kathleen, Jane, and Tara. I know so many people at our Moss Landing Thesis defenses comment on this, but I feel the same way. It's that we could really go into the front office whenever we needed support, needed some help. They were just great women to talk to, especially with Tara too. Tara is the graduate student coordinator. Tara and I kind of started in 2015 at Moss Landing together, and it's been so great to kind of settle into the community and kind of see where our careers are taking us together. A big thank you also to the shop guys, Chris, James, Billy, and Gary. They provided so much support to me, especially when I was doing my field work, helping me get the equipment that I needed. I really couldn't have done it without their support either. And then to all the professors like Jim, Mike, Gita, and Dai, who provided opportunities for me to grow as a better scientist and think critically about science, and taught those great classes at Moss Landing. I'm really appreciative of that. And everyone else, all you cool cats and kittens, not just at Moss Landing, but listening today, if you're here, you've likely provided some amount of support to me over the last 5 years. And I'm really grateful. And I wish we could be celebrating together. And lastly, I should mention, thank you to my funding sources. They're listed on the bottom there. Okay, I am done. Thank you so much for listening, and I'm ready to take questions.
All right, June, excellent job there. It looks like we have a hand raised from Mike Graham. So Mike, if you can unmute yourself, otherwise I can probably do it.
All good. Thanks, Scotty. June, that was awesome. But you kind of knew I was going to say it was awesome because it's so novel and so creative. I can't help but after listening to your talk, think of like herbivorous fish. Are they really running maybe a circular economy here, growing their own food on their own pee? I mean, that's like a dream of circular economists around the world to be able to do that. So I think it's really cool. But I wanted to, so here's my question, because there is a question, is what makes it even cooler, and your last graph where you were showing the yellow bar and how nitrogen from fish, is on par with internal waves, right? In certain aspects. A lot of those other bars were really loaded with nitrate. And as you know, ammonium is reduced. And for kelps, that reduction, that more reduced form is easier to assimilate. So in fact, on a per nitrogen basis, if you feed ammonium or nitrate to a giant kelp, it doubles its growth rate on ammonium. So those bars are even lower estimates as to the real impact here. So I was really struck, and I love your comment because I agree with it 100% from our aquaculture stuff. It's the persistence of this in the forest, the constant nature of the fish constantly peeing that probably really provides this incredible subsidy. So the question, what about those holdfast guys, right? I mean, that would be an incredible source of ammonium at just the right location to not be diluted out of the bed. I could just see that maybe based on some of your stuff, it might be a little bit more on the invertebrate side, maybe because of who inhabits it. But it seems like that would be a really cool mechanism for the kelps to retain a lot of that animal derived nutrients. And it's almost like a shot in the arm, right? They're going to get it directly into, do you know whether or not the holdfast can uptake? I mean, has anyone done anything like that where the actual physical inhabitation of the kelp or the seaweed is a way to get nutrients directly in?
Yeah, and I can see where you're going with this question too, because at Lindsay Cooper's defense just last week, where she was looking also at the storage of different nutrients in different parts of Pterogophora. So, and she found that result of having more nutrients within the holdfast part of the individual. So yeah, there's definitely a lot more space for doing uptake experiments and looking directly at the role of the consumers for the uptake rates with whatever species of algae. There was a study done fairly recently looking at the epibiont community in kelp forest in New Zealand, and they looked at the correlation with those epibionts that are attached to the actual kelp and the blade in different parts of the individual, and found that the growth for giant kelp was greater when there was a higher density of those epibionts. So I think there's definitely a lot to uncover there and could definitely be a whole nother thesis. So hopefully someone listening here will do some uptake experiments. Next question, please.
All right, so yep, Jen Cassell from UCSB, Pisco Queen herself has a question.
Hi, June. Thanks. That was amazing. And so nice to see all that hard work, come to not a conclusion, because there is so much more to do, as you said. Thanks for calling on me next, because actually my question builds on what Mike just said. And I think, June, you probably know what I'm going to talk about, which is, movement and movement of the animals. But I'll start with this question. So to me, the family level excretion rates, they didn't look that different. It may have just been, they were all smushed up on a plot. Maybe it was log, I can't remember. And so is the community-wide excretion differences that you saw using the Pisco data, would you say that that's really just comes down to biomass, or would have, big assemblage differences, family level or genus level, would that have changed anything in those community wide results? Because your plots about the MPAs look exactly like biomass plots.
Yeah, they do. I have some of those plots there. I think it's certainly close. Both are contributing. I think biomass is definitely a big part of it. Let me just see if I can pull up the relevant slides here to show and share my screen again. And I think, yeah, the idea that it's so driven by biomass, I think that this wasn't known before this research as well. So it's pretty cool to know, like, if there's more biomass in that area, there's probably more ammonium excretion also.
Yeah, it's still really interesting. And I don't know that you need to, I don't want to take up too much of the questioning time. I have one more quick one after this. So I'm not sure you need to share your screen, because I think it's just something to think about. And you have the data to play with that.
Yeah, and I did look at also, I didn't get a chance to talk about it today, but I did look at functional groups also. And so we see a lot more excretion from like the piscivores and the carnivores on the aerial scale. So some relevant for you, some of the effects from the marine reserves also, like the excretion from those groups was just increasing as they recovered from fishing also.
Yeah, cool.
So I think it's kind of all at play, and really the idea that those individual fish that can get to be a lot bigger and have more biomass frequently are the ones targeted by fishing.
Yeah, exactly. Can I just do one more quick one?
Yeah, sure.
So you measured excretion in the colder Central California waters and then in the warmer Southern California waters. Were any of the species measured in both places, the same species? And were there any differences in rates? So like, could these rates vary spatially? I hope the answer is no, but yeah.
Yeah, so yeah, a lot to uncover there. So I did not measure the same individuals, or excuse me, same species at multiple locations, just because on like Catalina, where I was sampling, the colder water species did not associate there. However, I have thought about this before, and I don't think that there would be a big difference between the excretion rates between the waters in Central California to the Southern California, because it's less than, like, it's a few degrees Celsius difference. It's under the 10-degree threshold, basically. Some other studies I was reading is if it changes by 10 degrees, then you see more of the metabolic effect changes, basically. So I think that this is just my kind of like back thinking about it, but it should be tested more. So I would think that if you're looking at a community, for example, like up in Alaska, if you're trying to characterize the fish excretion rate up there, it might be a little bit slower because the water is so much colder. But yeah, certainly more to be done. And I wonder if maybe there's even some sort of like conversion rate that you could, if someone did a study actually testing those differences, and then you knew how to just multiply it to account for the water temperature, that would be a cool direction to go also.
All right, so we'll go to George Jarvis, and then the other point, to Jen's question about biomass. Yes, it looks like biomass does a good job explaining excretion rates, but we didn't know until June's study was the actual number, right? How much ammonium is actually coming out of the fish. So at least we can now apply that. So, George, you're up.
Okay, June, very well done. Hi, it's awesome to see. Very cool results. I was curious about the second part of your talk with the sort of spatial component there.
Mhm.
And I was wondering like how localized these nutrients are, if there's potential for spillover. So for example, if you have, like you saw that in MPAs, there were higher nutrients in MPAs. And I was wondering if you looked at sites that were non-MPAs adjacent to MPAs, would they be more likely to have higher nutrients as well? And
Yeah.
as Jen was sort of getting at, it could be that things are more likely to move between, adjacent sites. But I was just kind of curious about that.
Yeah, I think spillover effects are definitely possible when you're thinking about the fish movements. For the uninitiated, maybe listening to this question, that just means the idea that you have these set marine protected areas, but a fish, it doesn't know where those boundaries are. So the fish might leave and go into an unprotected area. So I think that, yes, there could likely be spillover effects. Thinking about kind of the flow of nutrients, like in the water column itself, I think that's definitely another direction of research that more work could be done, is like, so the fish is excreting, how long does it stay within the different parts of the kelp bed? I imagine it would, again, stay right in there. I was also kind of thinking, yeah, I think different communities, there's so much to unpack there. It depends on which site you're at, because some of them, it might have different kind of like underlying currents and things moving around. I hope that answers your question, George.
Yeah, maybe it can't be answered. It'd probably be hard to tease apart, the nutrients moving versus the fishes moving.
Yeah, but I do
impossible.
think that honestly, so ammonium, like Mike mentioned, since it's the preferred source and so quickly utilized, frequently it's hard also to measure ammonium because you can get the ambient levels in the seawater, but then it's usually an underestimate because it's utilized so quickly. And also I talked a lot today about giant kelp, but there's also other primary producers utilizing ammonium also. And like, for example, phytoplankton use of ammonium sometimes exceeds that of giant kelp from some uptake experiments. So it's, I think there's a lot of constituents that want it in the ecosystem.
Cool, thanks.
Thanks, George.
All right, we got a question from Jim Harvey.
Great job, June. My question has to do with the I guess experimental design associated with it, and mostly this maybe I missed this, but I guess my question is related to, you did most of the experiments during the daytime, I think, with the assessments of excretion, and whether there's any change in excretion with day-night values, and therefore can you expand or use the daytime experiments as a representative of the entire day?
Yeah, good question, Jim. I do see studies with fishes and other ecosystems where they do report the excretion rates on a per day scale. And I think that as kind of a starting point, that it's okay to kind of multiply it up like that. I think there is potential that it could change. I can't say very definitively, but just thinking about like the fishes when they are hunkered down at night or something, they're not moving around. But my first guess is to think that it would be quite similar, just because, again, a lot of the nutrients are being released through their gills pretty much every time they're doing their respiratory processes. So I think it still would be quite consistent, unlike, you and me and people when we're like holding our pee when we're sleeping or something. It wouldn't work like that with the fishes.
All right, so, and just to follow up, just one last little thing. So having animals confined in a 5-gallon bucket during the experiment, do you think that's increasing or decreasing the possible excretion rates?
Yeah, there's actually been quite a bit of studies done also looking at like, what is the appropriate length of time to do these kinds of excretion rate experiments. And so I think that some of the studies where they looked at excretion rates over much longer periods of time, like 24 hours, 36 hours kind of thing, they found that that actually was not indicative of the actual excretion rates in the wild because of some fasting effects, even at just the 6-7 hour mark. It's mostly the effects of fasting is reducing the excretion. So with having my experiments be done in a 30 to 60 minute period, I think it's just indicative of how it would be in the wild. So fat increasing or decreasing, maybe slightly increasing, but if I had to pick one direction.
Okay, great. Thank you.
All right, we have a question from Laurel, and then we'll have after that a question from Amanda, and then I think we'll congratulate June. All right, Laurel.
Hey, June, good job.
Thank
I
you.
was curious, when you were looking at excretion rates inside and outside of MPAs, did you see that there was any like variability among the islands? Because, or I guess among MPAs, because they experience different fishing pressure over time.
Yeah, debating if I want to show that figure. So I did look at that. So the excretion rates were similar within an island, but there were differences depending on which island you were looking at when you're comparing them. And maybe I can just show this quickly in case there's someone that's really interested in this. You can look at this graph I'm going to show, but if not, then don't get overwhelmed. So I did look at trends, and so on the eastern islands of Anacapa and Santa Cruz, the excretion rates increased quite drastically, especially within the marine reserves. But then on the western islands, where the fishing pressure is not as strong, it did not increase quite so much. I don't know if you can, oh, there we go. So if you look like on this middle, this left column here, this is the total fish community excretion by island. So Anacapa is on the top and Santa Cruz is in the middle. So it's kind of going up their excretion patterns. Whereas on Santa Rosa and San Miguel, the western islands, it's not going up. And especially, so it's really, really strong marine reserve effects in the Santa Cruz Island. But we can talk more about that later or read my thesis.
All right, let's do Amanda as the last question.
Hi, June. I'm audible? I'm audible.
Yes, hi, Amanda.
Fantastic work. This is a really, really impressive, very cool project. I am always thinking about P, invertebrate P, and so it was really neat to see it from a fish perspective. And I'm wondering about kind of the individual variability within a species. So did you see differences within individuals of similar sizes? And did you sample any in Central and Southern California? And were there any differences there?
So the Central Southern California question, I think maybe that's kind of the same one Jen was getting at. So I did not, oh wait, you mean differences in excretion rates in Central California versus Southern?
For one particular species, say if you had looked at, gopher rockfish in Southern and Central California.
Right. So I did not look at the same species in two regions, how that might affect their excretion rates. I just used the one predictive one. But the first question, sorry, can you say it again?
Well, it's just thinking about how much variability.
Species variability.
Within species variability, correcting for size or not, for things that are similar sizes.
Right. So the species, the excretion rates were similar within a species. They were also similar to each other if they were within the same family, which I know isn't exactly what you're asking about, but I thought I'd just say anyway. And then, so what I was getting at with the part one results section, so even within a species, if you're doing the mass specific excretion rate, then it's the smaller fish that would excrete ammonium faster than the bigger one, just slightly.
Right. Okay. Yeah, thanks. I guess in my brain, I've been thinking about temperature as a factor. And with invertebrates, that increases their metabolic rate. So you'll end up with, more nitrogen excretion, or more of everything happening in warmer temperatures.
Yeah, so I think that likely would be at play with fishes as well, but I don't think the temperature difference between Central California and Southern California would be great enough to see a large effect of the temperature. But I think future studies could more explicitly test that.
Cool. Thanks very much.
All right, I think now if you have the opportunity to unmute yourselves, everyone can congratulate June, and then we'll move on.